Okay, hi everybody. This video is meant to give you extra guidance on how to navigate your way through the PowerPoint that is part of Lab 15. The overall goal of this lab is to take a look at two different areas of the desert in our environment and to compare the level of biodiversity of the plants in each of those two areas. Now, because of the format of our class being online, I'm not sending you guys out into the desert to do this. Instead, we're going to base our study on images that I have taken of two different areas using a drone around the Central Arizona College Signal Peak campus. And what this slide right here shows you is where those two areas are located. The first area is of the hillside overlooking the Signal Peak campus. And the second area is in this flat wash area that is at the base of the hill. So we have photographs, pretty detailed, high quality photographs of these two different areas. And we're going to be looking at those photographs and comparing the plant life in each of these two areas to determine which one is more biodiverse. And just to give you a preview of what those two areas look like, here's a shot of the hillside, and here's a shot of the flat wash area. Now, when you're doing this exercise, you are going to have to identify whether or not each plant is a tree, shrub, or a cactus. So there are three slides on here that will allow you to get a picture of what some of the different shrubs, trees, and cacti look like. You don't have to specifically identify what type of cactus or what type of tree or what type of shrub it is, but I do have that on here just for your information. These are also high quality photos, so you can zoom in here and get a closer look. For example, this little guy right here is a Palo Verde tree seen from above. You can recognize it because it's going to be pale green, it's bushy, it can be large, or it can be small depending upon how old it is. So before you proceed with the rest of the PowerPoint, make sure you browse and familiarize yourself with what I have on these slides, because that will help you make the determination of whether you're looking at a cactus, tree, or a shrub. And then the way that we're going to determine which of these two areas is more biodiverse is by using this concept that is used by ecologists called the Shannon Diversity Index. What you're going to be doing is looking at the images, the overhead images, and here I have superimposed a grid on those images so that you can systematically go through and look at each point on the grid and determine whether each intersection point has a tree, shrub, or cactus underneath it. It would be way too much work to count every single instance of a tree, shrub, or cactus in this picture here. And so instead, you can just go through and look at every grid intersection point and work your way systematically through this picture and this picture. As you're doing this, there are two ways that you can sort of tackle your tallies. You can have a piece of scratch paper on the side. So the way I've set up my scratch paper right here is area 1 is at the top. Area 1 is this picture right here. And so as you go through, you would tally every time you look at a grid intersection point and you see a cactus, tree, or shrub underneath it. And then this second row would correspond to area 2. Area 2 is this photo right here. And so let's do an example of this together. So I'm looking at area 1 right now. I'm going to zoom in and I'm going to start in the upper left-hand corner here of the picture. Actually, let's zoom in a little bit more. And I'm going to focus on the very first grid intersection point. You want to ignore the spaces in between the grid and just look at the intersection point. And I ask myself, do I see a particular plant underneath this grid intersection point? To me, I don't see anything, but I'm going to move down here to intersection point B, and it looks like this B1 is sitting on the edge of what's probably a Palo Verde tree right here. Okay, so now I'm going to go to my tallies, and I'm going to mark under area 1. I'm going to add a tally for one tree. Another way that you could do this if you're using a version of PowerPoint that allows it is there's a draw tab up at the top of some versions of PowerPoint, and you can actually take your drawing tool and you could say, okay, I've got this one, and I'm going to mark a T right here. There's a tree at this intersection point, and I've marked it there, and now I'm going to move on to the next one. Okay, C1. I don't see anything there. D1, okay, I see another Palo Verde tree, so I'm going to mark a tree here. And you keep going that way, marking whether or not, or tallying whether you see a tree, shrub, or cactus under each point until you have covered each of the grid points on this image right here. So when you get to the bottom of one row, jump back up to the top and go down the next one, and then keep going until you've covered the whole space. By the time you finish that, you will have completed all of your tallies for area 1. Then you move on to do the same thing for area 2. You go through and look at each intersection point and ask yourself, is there a plant underneath it? If so, which category does it belong to, cactus, tree, or shrub? Once you have your tallies, you're going to enter them into the chart that's on slide 14 here. So I'm just making these numbers up. Let's say for area 1, you found 3 cacti, you found 10 trees, and you found 7 shrubs. Okay, so that would give you a total number of plants that comes out to 20 total plants counted in area 1. You do the same thing here for area 2. And now the numbers that you come up with here are going to allow you to figure out which area is more biodiverse, but it takes a little bit of math to do it. The first step is calculating something called the relative abundance. Relative abundance, which we went over in the lecture, and we also, there's instructions about it in the lab document as well. Relative abundance is the proportion of a particular type of organism relative to or compared to the total number of organisms present. So if we want to calculate the relative abundance of cacti in area 1, you would take the number of cacti that you counted and you would divide it by the total number of plants. So on the next slide, it has a space for you to do that. The relative abundance of cacti in area 1, we go back to our numbers here. We saw 3 cacti in area 1 and 20 total plants. So the calculation would say, okay, I'm going to take 3 and I'm dividing it by 20. 3 cacti divided by 20 total plants, and that gives us a decimal value. The decimal value is going to be 0.15 for the relative abundance of cacti specifically. And you'll note on here that I've asked you to round to 2 places. Our decimal here is 2 places, so we don't need to do any rounding. But if you came out with a longer decimal, then please round it to 2 places. And you'll do the same thing for the trees, same thing for the shrubs. So if we look back, we had 10 trees out of the 20 total plants. And so that calculation would look like this. And that gives us 0.5. Basically, what this is telling us is that 0.5 or half of the plants in this area were trees, 15% of them were cacti, and then if we look at the shrubs, then this comes out to 0.35, 35% were shrubs. Okay, so we have the relative abundance of area 1. Of course, you'll need to do the same thing for area 2. And then once you have these relative abundance numbers, there's one final calculation to do to get to your Shannon diversity index. Shannon diversity index is going to come out with one number, and that one number will tell you which area is more biodiverse. So once you've completed the calculations on slide 16, then slides 18 and 19 are the final boss here. So for step 1, you're going to take the relative abundance value that you calculated in slide 16. And by the way, this slide right here, 18, this one pertains to area 1, and this one pertains to area 2. So you have two separate slides, one for area 1, one for area 2. So for area 1, if we look back at slide 16, the relative abundance of cactus, trees, and shrubs. For cactus, it was 0.15. We're going to put that right here. For trees, it was 0.5. And then for shrubs, it was 0.35. Okay, so step 1, check. We've retrieved our values from slide 16. Step 2, use your calculator to take the natural log of this value. Now, natural log is a function that you can find on any scientific calculator, and if you don't have a calculator available to you, then all you have to do is go to Google, and if you type in scientific calculator, it will pull up a scientific calculator where you can plug in numbers. And the natural log function in the scientific calculator is represented with ln, which is log natural. So if we put in ln, and then we type in our relative abundance value, let's just do it for cactus first. We've taken the natural log of 0.15, and we hit equals. Okay, now we have a number here. And we're going to put that number in this column. And as we can see, it is a negative number, but that's okay. We're going to, let me get rid of that. We're going to round to 2 decimal places again, which ends up rounding to negative 1.90. And then the last step here is we will take this number right here and we will multiply it by this number. And that's what gives you what goes in this final box here. So if we take 0.15 times negative 1.9, and we can do this in our scientific calculator again. Then we get the number 0.285, which rounds to 0.29. Okay, so we've figured out this value for cacti. We're going to do it for trees, and we're going to do it for shrubs as well. So again, we would take this value right here. We would use our scientific calculator to take the negative log of that value, and that goes in this box. Then you multiply them together, and you get your final answer here. And then you do the same thing for this last one here. So quickly, I'm going to do these ones as well as an example. The natural log of 0.5 is negative 0.69, rounded to 2 decimal places there. And then when we multiply negative 0.69 times 0.5, so this one by this one, then we get negative, sorry, I forgot to put my negative value right here, negative 0.35. And lastly, we're going to do the same thing for this one. We are going to take the negative, or the log of 0.35, and that gives us the value of negative 1.05. And then we take negative 1.05 and we multiply it by 0.35. So this column multiplied by this column again. And that gives us negative 0.37. Okay, so we've completed those calculations. As you can see, step 4, add these three numbers together. Add the three numbers in the last column together. And then step 5, multiply by negative 1. Basically, all this does is it changes the negative value to the positive value. Whatever we get in this column is going to be negative, but we don't want it to be negative. We want it to be positive, so we basically just flip it to a positive number. So if we take negative 0.29 and we add that to 0.35 and we add that to 0.37, that would give us negative 1.01, but we flip it to a positive number. So our total number is 1.01. So now you have your Shannon index value. This is the number that ecologists use to determine biodiversity. And now you've done it for area 1. You would do the same thing for area 2 on the following slide. So you'd start by getting your relative abundance values from slide 16. You drop them in here. You take your natural logs. You multiply these two columns together, and it gives you this final column. You add everything you got in this final column together, and you flip it to a positive number, and then you get a number here. And then you're going to finally determine which number is bigger. Is the number for area 1 bigger, or is the number for area 2 bigger? Whichever one is bigger is the area that has a higher level of plant biodiversity as we have measured it. And remember, that's ultimately what you're going for in this lab, is to figure out which area has greater biodiversity. So then you'll want to save this PowerPoint with the numbers that you've input into it, and you'll upload it back to Blackboard as part of the assignment and answer a few additional questions, and that will be the end of this lab.